Hey guys, I wanted to go over and give just a few instructions on this assignment for today. So if we look here at the background, it says a recent agricultural report suggests that a fungal pathogen, so it's a fungus, so that's the scientific name, R. anaerobis, may negatively affect the growth of soybeans, and this is the type of soybeans, glycine max. Soybean growth decreased during 3 years of high rainfall, and the soybean roots were poorly developed. Close relatives of that pathogen are plant pathogens that grow in the soil. A lectin-like protein was found in the soil around the roots. This protein may have been secreted by the fungus, so that's important. The protein is coming from the fungus. Lectins induce mitosis in some root apical meristem tissues. Meristem tissues are just a plant type of tissue. In many instances, rapid cell divisions weaken the plant tissue. So we're going to investigate, here's our investigation, whether the fungal pathogen lectin affects the number of cells undergoing mitosis in a different plant using the root tips, okay? So you have to make a hypothesis. This is your experimental hypothesis. And then you also have to make a null hypothesis. Remember, we talked about what a null hypothesis is, okay? Null hypothesis, no significant difference between the experimental group and the control group. So then you have to identify your variables. You would write out just a few little procedural steps here, what you would do to investigate this, okay? So something like, having some root meristems from the soybean plants, treating some with lectin, treating some without lectin. Basically, that's what we're going to do. And then you would look at them if we're trying to see if this lectin affects the number of cells undergoing mitosis. Then after you treat them with lectin, you'd basically just look at them under the microscope and count the stages, kind of like you did yesterday in your assignment. So the data has already been collected for you. And so we have our control group. So this has no lectin. And then we have the ones that are lectin treated. Now, you can ignore this part for right now. We'll address that later. We're just looking at the first two rows here. So control and lectin treated. And they've counted several cells. Notice the cell number totals are different, so they didn't count the same number of cells each time. So that's fine, but you'll want to record them as a percent to be able to compare them to one another, because essentially what we're going to do is chi-square. And you can't compare the values with each other if your totals are greatly different, okay? So, you are going to do chi-square on this data to answer the question, does lectin affect the number of cells in mitosis? Does it induce mitosis? Meaning, does it bring it out of interphase? Okay, are more in mitosis than in interphase? And so there's a few little directions for how to do that, okay? You've got a space for your calculations, and you should have this on paper. I want you to show your work here, okay? I want to see what you have done. Don't do this on your head or anything like this. Show your work for chi-square. And then you're going to write a brief analysis, okay? Stating whether you accept or reject the null hypothesis and what that means. We have done several chi-squares now. You all should be able to do this. I will tell you for this, you only have two things that you're comparing. You're comparing interphase cells to mitosis cells. So you're going to add up your prophase, metaphase, anaphase, telophase as your mitosis cells. That will be one group, and that will be two groups. So in your chi-square, your degrees of freedom will be 1. There's two groups or two categories minus 1, so your degrees of freedom are going to be 1. If you're not really comfortable with chi-square still, go back and look at the M&M lab again. You should have that in your Unit 1 stuff. Look up a YouTube video about chi-square. Go back and rewatch where I did M&M chi-square. You've got to be good at chi-square. I can promise you, you will have to do chi-square. If you do science in college, if you take the AP Biology exam, which I'm signing you all up for today, actually, you have got to do chi-square. So let's just knock this out here. So you're going to write your analysis, and then There's one question here at the end. How does different amounts of caffeine affect cell division? Okay, and so that's where you're going to use this part. Okay, and you just can write a 1 to 2 summary sentences explaining the effect of caffeine on cell division. So if you all have any questions with this, if you work through it and you're not sure about it and you want me to check it, I'm happy to do that. Just, text me or email me a picture of it and I'll look over it and say, yeah, you got that right, go ahead and submit it, or no, let's think about this and go back to the drawing board on that, and I'll try to help guide you through it. But this is an important assignment because it is another way for us to practice chi-square. So happy Friday, and y'all have a great day.